Assalamu alaikum. This is Dr. Aisha Zulfikar, and the topic we are discussing today is gluconeogenesis. Gluconeogenesis is synthesis of glucose from non-carbohydrate compounds such as lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. These are known as major substrates or precursors for gluconeogenesis. They are usually asked in your SP and Viva that what are the precursors for gluconeogenesis. So they are lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. Glucose can be synthesized from all of these compounds. Now, site where does gluconeogenesis occur usually in our body? So, gluconeogenesis occur mainly in our liver. To a lesser extent, it can occur in renal cortex. The pathway is partially mitochondrial and partially cytoplasmic. It means some of reactions take place in mitochondria while other reactions take place in cytoplasm. And about 1 kg of glucose can be synthesized by gluconeogenesis every day. You might be thinking what is need of gluconeogenesis when we have glycolysis and when we take like glucose. There are times when our body runs out of glucose, and you know our body requires continuous supply of glucose for its functioning, for generation of ATP. So in the absence of glucose or in the absence of glycogen stores, our body converts these non-carbohydrate parts or compounds into glucose to meet the needs of the body. Now, what is important of gluconeogenesis? Brain, CNS, erythrocytes, testis, and kidney medulla are dependent on glucose for their continuous supply of energy. Human brain alone requires 120 grams of glucose every day. Glucose is only source that supplies to the skeletal muscles during anaerobic conditions. So see, all of these functions require glucose. Continuous supply of glucose is very much essential, or you can say complementary for body to function normally. Now during starvation, gluconeogenesis maintains our normal blood glucose levels. The stored glycogen is depleted within first 12 to 18 hours of fasting. See how quickly this glycogen stores are depleted, are utilized just in 12 to 18 hours of fasting. On prolonged starvation, gluconeogenesis is speed up and protein catabolism provides a substrate, namely glucogenic amino acids. So as you have heard, first glucose burns and protein. So this is how glucose is provided to our body continuously. Now what are reactions that occurs in gluconeogenesis? Actually, gluconeogenesis is the reverse pathway as glycolysis. As you have seen in my previous lecture, the reactions which take place in glycolysis, when you will reverse those reactions, they will form, it is known as gluconeogenesis. We will see all these steps in detail. The three irreversible steps of glycolysis are catalyzed by the enzymes hexokinase, phosphofructokinase, and pyruvate kinase. These were irreversible reactions. So these will not occur in gluconeogenesis. They are the irreversible reactions of glycolysis. Irreversible means they cannot be reversed. So these three enzymes and these three steps will not occur in gluconeogenesis. These three stages are bypassed by alternate enzymes that are specific to gluconeogenesis, and those enzymes are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. So these are the enzymes which are different in gluconeogenesis, which are not present in glycolysis. So first of all, pyruvate. We are talking about this first substrate, pyruvate, which is the product of glycolysis. This pyruvate is converted into phosphoenol pyruvate. This occurs in two steps. Pyruvate carboxylase is a biotin dependent mitochondrial enzyme. Biotin dependent means biotin acts as a coenzyme for pyruvate carboxylase to function normally. It converts pyruvate to oxaloacetate in the presence of ATP and carbon dioxide. This occurs in mitochondria. This enzyme regulates gluconeogenesis and requires acetyl-CoA for its activity. So, this is a regulatory step in gluconeogenesis. So, pyruvate is converted into oxaloacetate, and this is oxaloacetate is synthesized in the mitochondrial matrix. Now, this oxaloacetate is transported to cytosol. Due to membrane impermeability, oxaloacetate cannot cross mitochondrial membrane. So, there is a reversible conversion of oxaloacetate to malate by MDS, this malate dehydrogenase enzyme. It is present in mitochondria and cytosol both. Once this oxaloacetate will cross the mitochondrial membrane as malate, it will be again converted into oxaloacetate. In cytosol, phosphoenol pyruvate carboxylase converts oxaloacetate to phosphoenol pyruvate. So, this is how it occurs. This is mitochondria, this is cytosol. So, this is mitochondrial matrix, and this is oxaloacetate. So, pyruvate is converted into oxaloacetate. Now, this oxaloacetate needs to transport to the cytosol for further functions to occur. So, there is a reversible reaction, reversible conversion of oxaloacetate into malate in the presence of enzyme malate dehydrogenase. Now, this malate will cross the mitochondrial membrane and move to cytosol, where this malate will be again converted into oxaloacetate. So, this whole thing occurred just for the transport of this oxaloacetate to the cytosol. Now, this is again a diagrammatic illustration of the reaction. This pyruvate is converted into oxaloacetate in the presence of pyruvate carboxylase enzyme, and biotin acts as a coenzyme. This is in mitochondria. This oxaloacetate is converted into malate, then again converted into oxaloacetate in cytoplasm. This oxaloacetate now is converted into phosphoenol pyruvate in the presence of phosphoenol pyruvate carboxykinase enzyme. Now, we will see what happens after this PEP is formed. There is another thing in this, that we saw that lactate also acts as substrate for gluconeogenesis. So, this lactate is basically, this lactate is converted from pyruvate in the anaerobic respiration, but they are interconvertible. So, when there will be low amount of glucose in our body, this lactate will convert into pyruvate, and this pyruvate will again convert into phosphoenol pyruvate, and then gluconeogenesis will take place. Now, phosphoenol pyruvate undergoes reversal of glycolysis until fructose 1,6 bisphosphate is produced. Okay, all these steps will occur in reversible form until the formation of fructose 1,6 bisphosphate. The enzyme fructose 1,6 bisphosphate converts fructose 1,6 bisphosphate to fructose 6 phosphate, and it requires magnesium ions. This is also a regulatory enzyme. This is the second regulatory enzyme after pyruvate kinase. Now, this is showing, this is glycolysis, this side is glycolysis, pyruvate was formed. Now, we will see how gluconeogenesis occurs. This pyruvate is converted into oxaloacetate in the presence of pyruvate carboxylase enzyme. This oxaloacetate is converted into phosphoenol pyruvate in the presence of phosphoenol pyruvate carboxykinase enzyme. This PEP or phosphoenol pyruvate is converted into 2-phosphoglycerate enolase. This is the same enzyme. This is interconvertible or reversible reaction. This was present in glycolysis. Now this 2-phosphoglycerate will be converted into 3-phosphoglycerate in the presence of phosphoglycerate mutase enzyme. Now this 3-phosphoglycerate will be converted into 1,3-bisphosphoglycerate in the presence of phosphoglycerate kinase, and this will be converted into glyceraldehyde 3-phosphate in the presence of glyceraldehyde phosphate dehydrogenase. So now this will be converted into fructose 1,6-bisphosphate in the presence of enzyme aldolase. Now there is some difference. This fructose 1,6-bisphosphate will be converted into fructose 6-phosphate in the presence of fructose 1,6-bisphosphatase. Basically, this phosphatase breaks down one bond and separates one phosphate from fructose 1,6-bisphosphate, which had 2 phosphate has now 1 phosphate, that is at 6th position. So fructose 6-phosphate in the presence of fructose 1,6-bisphosphatase enzyme. Now this fructose 6-phosphate will be converted into glucose 6-phosphate in the presence of phosphohexose isomerase. This is also a reversible reaction. This was not a reversible reaction, hence this is regulatory step. This is also not reversible reaction, hence is it regulatory step. So now this glucose 6-phosphate will be converted into glucose in the presence of glucose 6-phosphatase. This phosphatase will break down this phosphate bond and separates it from the glucose, and free glucose will be delivered into the blood. All the arrows which show purple and light blue color, they are reversible reactions. They are same in gluconeogenesis and glycolysis. Where these both arrows are absent, these are different reaction, irreversible reaction, and enzymes are different in gluconeogenesis and glycolysis. So this was whole summary of gluconeogenesis. We have already discussed it that glucose 6-phosphatase catalyzes the conversion of glucose to 6-phosphate to glucose. It is present in liver and kidney but absent in muscle, brain and adipose tissue. Liver can replenish blood sugar through gluconeogenesis. Glucose 6-phosphate is mainly present in the liver. Okay. Now we are done with how this glucose is formed from lactate and pyruvate. Now we'll see how this glucose is formed from amino acids. The carbon skeleton of glucogenic amino acids, all except leucine and lysine, they result in the formation of pyruvate, are the intermediate of citric acid cycle, which ultimately results in the synthesis of glucose. So this is this is muscle, this is liver, and this is alanine. This is alanine, a glucogenic amino acid. It goes to the liver. It is converted into pyruvate. Ammonia is released, which is converted to urea. This alanine is converted to pyruvate. Then pyruvate is converted into glucose by all the steps we discussed earlier in gluconeogenesis. So this glucose will go in the blood and go in the muscle, and then it will be again converted into pyruvate. Muscles will use this glucose for energy, for like walking and for everything, for all the activities we do. So this glucose will be converted into pyruvate, then pyruvate will convert into alanine, alanine again go into the liver, and then glucose. This is the cycle. This goes on when there is absence of glucose stores. Now glucogenesis from the glycerol. How glucose is formed from the glycerol. Glycerol is liberated in adipose tissue by the hydrolysis of fats, or triacyl glycerols. So glycerol is the end product of fats metabolism, or fats are fat metabolism. The enzyme glycokinase, this is found in liver, kidney, absent in adipose tissue, activates glycerol through glycerol 3-phosphate. It is converted into dihydroacetone phosphate by glycerol 3-phosphate dehydrogenase. This DHAP is an intermediate in the glycolysis. When we will go through the glycolysis, you will see this DHAP is intermediate in glycolysis. So, this is a fatty cell. This adipocyte, this triglyceride is broken down into fatty acids and glycerol. This glycerol go in liver. Because the enzyme which is required for its conversion is not present in adipose tissue, it is present in liver. So, this glycerol will be converted into glycerol phosphate, and then it will be converted DHAP, which will be converted into glucose. This glucose will go in the blood and will circulate in the blood and go into different parts of the body where required. So, we are done with gluconeogenesis from pyruvate, from lactate, from amino acid, from glycerol. Now, see how glucose is formed from propionate. Oxidation of odd chain fatty acids and breakdown of some amino acids such as methionine, isoleucine yields 3-carbon propionyl CoA. Propionyl CoA carboxylase acts on this in the presence of ATP and biotin and converts it into methylmalonyl CoA, which is then converted into succinyl CoA in the presence of vitamin B12. Then this succinyl CoA formed from propionyl CoA enters gluconeogenesis. So, this propionate not directly forms glucose, but it is converted into succinyl CoA, and then the succinyl CoA enters gluconeogenesis. Now, gluconeogenesis from lactate, we have discussed earlier, but this is a little bit in detail. This is in a special cycle known as Cori cycle. We will see it. Now, to prevent lactate accumulation, body utilizes Cori cycle. I told you that when there is a lot of lactate produced from pyruvate, it can cause lactic acidosis. Now, to prevent that lactate accumulation, there is a cycle known as Cori cycle. This lactic acid from muscle diffuses into blood. It reaches liver where it is oxidized to pyruvate, and then it enters into gluconeogenesis. So, this is how this lactate is converted into pyruvate and then into glucose. Now, this regenerated glucose can enter into blood and then to the muscle again for energy production. So, this cycle is called Cori cycle. I will show you how this is a cycle. Okay. So, there was glucose. This glucose went to the muscle, then there occurs glycolysis occurs in the muscle. This glucose was broken down into pyruvate, and in anaerobic respiration it was converted into lactate, and now this lactate will go into the liver when there is a lot of lactate or when there is low glucose levels in blood. This lactate will be converted into pyruvate, and this pyruvate will enter gluconeogenesis. It will form glucose, and this glucose will go again to the muscles via blood, and it will seeing the cycle will repeatedly occur. So, there will be no accumulation of extra lactate in the blood. This is very important cycle. This usually comes in your SP, this Cori cycle. So, you have to remember that how this whole cycle occurs. Now, this is glucose. When when our body eats like when we eat a meal, heavy carb meal, our body releases insulin. Okay. And when we are starving, our body releases glucagon. I am so sorry. Now, this glucose, it depends upon how much glucose is present in the blood, that what pathway will carry on. When you have taken a meal, when there is a lot of glucose in your blood, then there will be glycolysis, and whereas when you are starving, when your blood glucose levels goes down, then there occurs gluconeogenesis. So, glycolysis is favored by insulin, And gluconeogenesis is favored by glucagon and glucocorticoids, whereas it is inhibited by insulin. When we are full, when there's enough glucose in our blood, then there will be a lot of insulin. So this insulin will prevent further formation of glucose from gluconeogenesis. So this is the simple hormonal regulation of gluconeogenesis. This was all about gluconeogenesis.